So Jill Gallaher is an applied research scientist in the Department of Integrated Mathematical Oncology at the Moffitt Cancer Center. She has a PhD in biomedical physics from East Carolina University, and her research focuses on a variety of questions related to tumor biology and treatments. In particular, she works on models to exploit evolution to treat heterogeneous tumors, define heterogeneity in glioblastoma using multi-scale data and models, uncovering mechanisms for immunotherapy resistance with imaging, and comparing optimal strategies for cell and fluctuating hypoxia. So we're really thrilled to have Jill here to deliver the second keynote talk of the workshop on agent-based modeling in glioblastoma. So thanks, Jill, and we can see your slides.
Okay, good. Cool. Well, thank you so much for inviting me. It's been a really great conference so far. About 10 minutes ago, they started chopping up the sidewalk with a circular saw. So if it gets loud, just let me know. I'll speak up. But I think it's done anyway. So we've had a lot of talk about agent-based models today, and then a little bit today and yesterday about glioblastoma. So I kind of wanted to start this off to talk about, you know, every model is kind of formed by the question, and the complexity level is determined by the question that you're trying to answer. In this case, I think it's also fair to say that it's determined by the people that are involved and the perspectives that they bring in. So I wanted to start out kind of defining where this whole perspective is coming from. And this comes from a grant from the McDonald Foundation that started this whole collaboration that we call Bonk, the Brain Oncology Network of Knowledge, headed by Kristen Swanson's lab, who kind of brought a lot of the continuum modeling and also collecting this huge database of imaging, clinical images of glioblastoma. And also the Canal lab. Peter Canal is a neuropathologist. He also does these great experimental models. And then the Anderson lab. So Sandy Anderson is who I work with, and we do the agent-based models. And so bringing all these perspectives together, you know, at times it can be aggravating. We're all kind of bumping heads on trying to find the consensus of how complex or how simple we might be on certain ideas, but also, you know, in the end, it becomes stronger. And so this is one of my favorite things, is bringing all these people together and coming up with a common language and a common idea about how to model these things. And so this is kind of where this perspective is coming from. So glioblastoma comes from these glial cells in the brain. Glial cells form these diverse cells that have all sorts of different functions, from the ependymal cells that contact the cerebrospinal fluid, the astrocytes that connect the neurons with the blood vessels, the oligodendrocytes that insulate the neurons and allow like huge you know, distances traveled of an electrical signal all the way from the brain to your left toe, and your microglia, which are like immune cells in the brain. But the glioblastoma is generally arising from these two cell types. And so we have a certain viewpoint into the disease, and it generally comes from imaging and from H&E and biopsies. What we generally see is MRI scans that give this T1 weighted image, which is more like a density of the brain tumor, and then the T2, which looks more about this swelling and edema that occurs around it and might pick up some of the cells that travel quite far from the core of the tumor. In the H&E, we saw earlier how it can form this pseudopalisading necrosis. So the cells start to die, and you get these cells that line up around the dead portions. And then there's all sorts of molecular markers that not only inform what the tumor might be doing, but also how it might respond to treatment. We're really concerned with, you know, the basic structure of growth and of the tumors, which really depend on how fast they're proliferating, how fast they're migrating. And the brain structure, specifically the gray and white matter, really influence this. So of course, as we've heard from Thomas Hillen yesterday, he was talking about how this white matter, it's called white because these are fatty sheaths that are white in color, and they allow this insulation of the brain neurons. And so basically they're these huge, they almost provide like pathways, highways that the cells can travel along and influence migration and speed up migration. And along those pathways, there's also really large blood vessels. But these large blood vessels existing in the white matter then trail off into smaller and smaller capillaries eventually. And so you get a lot of vasculature in the gray matter that feeds the tissue there with a little bit more oxygen and blood. And in the end, with glioblastoma, treatment is really difficult and recurrence is probable. I think the 5-year survival for glioblastoma is about 7%. So 2-year survivors are considered long-term in this disease. And there's a variety of reasons, one of them being how difficult the treatment is. It's just surgery, you can't remove everything. There's very important anatomy that you need. You can't just cut out. The blood-brain barrier is another issue, that drugs can't just cross over this barrier that is there for a reason. So this limits the kind of drugs that you can give patients. And then, targeted therapy or even, chemotherapy can only be possibly attacking a subset of cells. And so you have this recurrence. And so that's kind of the part that we really wanted to study there. And so the question that we were asking here is, how does tumor heterogeneity and GBM affect growth and treatment response? So when you look at GBM through this kind of continuum model set of eyes, you can study it as kind of a traveling wave using this reaction diffusion model. And so you can look at how the density of these cells can then be translated into these images that you see. But ultimately, growth and even a lot of treatment is pretty predictable by these PD models, but it's really the longer term post-treatment dynamics that become quite varied, and you start to get a lot of differences between patients. And the issue is that you have selection. So chemo, hits the fast proliferators, so it's selecting for slow proliferators. Same with radiation, only, it also depends on hypoxia. Anti-angiogenics, these actually select for faster migrators. So what we did then is take this experimental model, and what we want to do is really observe single cells in the context of the brain in order to get a better idea of the heterogeneity that actually exists and where it's coming from. And so this experimental model, the way that it was done, it's basically what they do is they inject a retrovirus that incorporates into the cell's DNA, and it forces them to overexpress this Pdgf, this platelet-derived growth factor. And in doing so, it releases this Pdgf into the brain tissue, and it stimulates not only themselves, it self-stimulates these cells, but it also stimulates all of these progenitor cells that are lying in the tissue. They're basically waiting around for an injury, and they help with the wound response. So in doing this, this overexpression of Pdgf actually stimulates not just the cells that were originally infected, but also all these tissue cells that are just sitting there waiting. But what's nice about it, it's really, they can track these single cells. So what they do is they cut this tissue So it's about 300 microns in height. And they let it, they put it in some solution so that it can stay alive for a little while, and they just watch these cells. So they're tagged with GFP and DS red, and they can just watch them over time and see how they're migrating and proliferating. And so this was originally just to see how this recruitment occurs. Susan Massey in Kristen Swanson's lab did this continuum model, these PDE system, to kind of look at how your infected cells and recruited cells are interacting along with this PDGF in a 1D model. And it worked pretty nice to see how you can see how much of these cells are recruited. So you've got about 80% of these recruited cells, and only about 20% are these infected cells. And so kind of just observing this in one dimension, it works out pretty well. And you can then figure out what some of these parameters are. So we can start there to, why am I not moving here? Hold on, slide stuck. Can you still hear me?
We can still hear you.
Okay. Escape's not working either. What's happening? Oh, here we go. We're good.
Great.
Okay. So, so this is the large scale dynamics here. We've got these two types of cells in PDGF, but really to capture this underlying heterogeneity, we need to match not just to the larger tissue scale dynamics, but the single cell data. So this is the basic idea, is taking into account this multi-scale data, the imaging data, the tissue data, the cell type distribution, but also the individual cell tracks. And so we take these scales and find similar metrics that we can use to compare in this agent-based model. So we can extract imaging data and tumor size, but we can also look at the ratio of different cell types, and we can look at the individual cells. And so something important to note here is that when we talk about the environment and this PDGF that's influencing the environment, we have this kind of potential phenotype. So each cell has kind of a potential, but it might not ever realize that potential because now it's in an environment, but in 2 minutes later, it could be in a different environment. So you have what the cell could possibly do and what the cell actually does and how it comes out to be measured and the metrics that are defined by that. And so that's one theme that will keep coming up. So yeah, imaging data, this was already determined from Susan's paper. And then we have the ratio of these two different cell types at 17 days post-infection. And so this is just a biopsy in the core that we can match to. But the new stuff really here is the single cell data. And so here's a little movie of what this looks like. So as this tissue is kept alive, they're watching all these cells move around in the tissue, and you can see these are brain cells. They're not these, nice little neat cancer cells that's always depicted in your agent-based models. Just something to consider. They're moving around. You can see they're almost sensing their environment. But another important thing to consider is that they are responding to this environment. I'm just going to play it once more. And that this gray matter tract, you can't see it there, but it kind of goes from the top left to the bottom right. And it is providing this highway where the cells are moving in both directions along that pathway. And so this is kind of my attempt to put all of the information from those movies into one graph. And so it may be a little bit much to look at first, what you'll see here in the background is a density plot of all the cells. And so the grayer it is very dense all the way to white where you have no cells. And then on top of that, you see these cell tracks. So the recruited cells are in red. They get more yellow if they're moving faster. The infected cells are in green and they get more blue if they're moving faster. And I've placed 2 arrows on there to indicate the mean direction of where they're moving. Anytime there's a cell division, you can see an asterisk. There wasn't a whole lot of correlation with the environment or the other cells around to indicate where those were happening. But then these cells also stopped for long periods of time. So anytime they stopped for an hour or more, I placed a circle there in proportion to how long they stopped. So it's really complicated movement. We can look at it in a more detailed way, or we can look at it more simply. In this case, a little bit more simply, we noticed that since this movie was only 25 hours long, or these movies, there were several of them that we looked at, we found that the cells only divided about half the cells divided in that time. So we couldn't really find any time where the cell was, born and then divided. It all was, we had to define this as a percentage of divisions per hour. But even using that metric, we did find that the recruited cells proliferated more at both 2 days and 10 days. And then we can look at this wind-rose plot where it shows if you start all cells from a single 0.00 and allow them to go out, you kind of get this sense of how they're moving, and look at the mean squared displacement over time. And you see that these recruited cells do move a little bit faster. But then we can also look at this detailed movement behavior. So the way that we did this here is if a cell is moving, it might be moving, but if it doesn't move more than 5 micrometers, we consider it to still be stopped. But then we have this run if it doesn't stop, or if it doesn't change angles of more than 15 degrees. And so doing that, we quantified the stop times and the go times, and we found that these cells actually stop a lot more than they move. They move in these little spurts and they stop for a long time. Doing that, we can find in this graph here that there's a variety of speeds. So if we just quantify how fast they're moving when they're moving, we see that there's this variety of speeds. And then that's mean over the whole time, while this graph over here is showing the infected recruited. This is the instantaneous speed, so how fast they're moving for each run, and then the turning angles that we've got here. But this is a lot. If we want to actually fit this data to the model, we had to define something a little bit simpler. And I know this seems a little too simple, but it's different than if you're looking at a PDE and you want to determine displacement because you're defining a diffusion rate. This isn't that. This is the total distance traveled. So the cell could be moving all about and not really go anywhere. And so we found that this was actually quite a good metric to use for both the model and the data. And here we find that still the recruited cells have a little faster average speed, and also the divided cells. So anytime a cell divides, what we saw is that it kind of stopped for a second and then shot off in different directions. So the minute that a cell is born, they tend to go off in opposite directions, and that made it so that their speed was a little bit faster. And so the way that we then put this model together is using a hybrid agent-based off-lattice model with a diffusing growth factor. We imported this brain atlas from the scalable brain atlas and defined a white and a gray matter, just in the plane that we had for the data. We define the white matter and the gray matter such that the carrying capacity due to that vascular influence and the persistence due to the white and gray matter reflected what we saw, what we know from what happens there. We can define density on a larger, coarser grid, and as the cells get dense in a certain area, the cells will then go into quiescence and reduce their migration. Then for each cell type, you've got these infected cells that are secreting but also consuming Pdgf, and these recruited cells that are just consuming Pdgf. And we can observe, the Pdgf and the phenotypes. And this is the way that the model flows here. At each time point, we calculate the density of cells. We split them into cells that are inactivated and activated. So there's a field of these recruitable cells that exist and kind of lie in wait to be activated. But if there's no Pdgf, they're just kind of sitting there. So we make sure that we're not trying to calculate what they're doing all the time. If they are activated, if there's enough, if it's quiescent, if there's not enough space, then it just kind of moves to the next cell. But if it's not, then we check to see if it's ready to divide, and if so, it divides. And otherwise, we update the migration and we update the Pdgf consumption and secretion. And after all the cells, we go through all of that, we do the Pdgf decay and diffusion. I got stuck on a slide again. Here we go. And the Pdgf dynamics, we decided to do this on a hexagonal lattice. It was a little bit challenging to get going, but I think it kind of, you get less artifacts that happen with some of these, grids, but you have to just be careful with indexing. But otherwise, it's pretty straightforward to calculate how diffusion is happening on that hexagonal lattice. And then the last thing is cell heterogeneity. So like I said, you have each proliferation rate and migration speed is determined by an intrinsic potential of a cell and then its response to the environment. So for the infected cells, they not only have the environmental response, but they have an autocrine stimulation. So because they're producing this Pdgf, they keep some inside the cell and self-stimulate. For the recruited cells, we included this factor beta, which modulates that K, which is kind of like in a way that it is less than 1, so that it responds faster to a lower amount of Pdgf in the environment. So you can see down below how those parameters affect the response function. There's quite a bit of model inputs. We decided on 16 and defined their ranges, everything from tissue parameters, Pdgf parameters, and the response, and also, things that were changing in the model, such as the proliferation and migration and heterogeneity in those. The outputs here are just all of those things that we can measure, such as tumor diameter, the ratio of cells, and the average proliferation rate and migration rate and standard deviation at these 2 time points. Okay. So the last thing to consider is that in these agent-based models, especially with a lot of migration, you don't have a nice clean tumor diameter. And so when we do measure the diameter, we're constantly finding the center of mass and defining angles around this to see where the density is at each angle, and kind of finding the maximum that exceeds a certain threshold. What I'm showing in this picture here on the right, it's at 50%. We ultimately changed it to a threshold of 10% of the carrying capacity because that seemed to do best. Just one last, it's just one last metric that you need to define on the fly to be able to easily go through a lot of simulations and match metrics. And the way that we went through this estimation of parameters is, I tried a lot of things. I tried simulated annealing. I tried just, random sampling. And I ended up with this kind of hybrid approach. And the way that I did this is basically you take a bunch of parameter sets. I'm throwing this on our cluster. So I've got like 5,000 simulations with different parameter sets. I run this through the model and define the errors for each simulation. And so this is basically defined as a normalized weighted least squares, where the weight is basically determined by however, since we're comparing things like tumor size over time, we're comparing a ratio of cell types and speed and proliferation. There are all these different types of measurements. And so each one is weighted and to the amount of margin that we'd like. Then we sort these so that you have the least error that's shown there on the right. We have selection, so we take 10% of the least error and just take that down and redefine our next iteration with that one just comes to the next iteration. And then we take that cohort, that selected cohort, and have some mutation within it. And then we define these, we define however the distributions come out, then we resample from that to determine the last 50%. So the reason why this worked out is because, with a large amount of parameters that we're trying to estimate, there are some correlations between these. These are not completely independent parameters. So the selection actually preserves that. The mutation allows you to get a little closer to what closer to a better solution. And then the resampling allows you to still get at a global, a global minimum there. And so that's the reason why it kind of came up that way. And then this probably went through 5,000 parameter sets. It probably went about 10 to 12 iterations. So yeah, it was interesting to hear Paul talk about, it seems to be maybe on the same order when you're doing the machine learning. So anyway, so it's something that worked for me. I'm trying to write this up and into a Jupyter notebook, and maybe it'll be useful for somebody. So what we did first is only fit this to the size dynamics. So if you look in that yellow box, you can see the size dynamics are fitting really well. Everything else has not been fit. And you see there's a broad range of parameters that this lands on. If we just look at these results, it's really varied what we get. And so here's a video of what you can see, kind of a variety of different solutions. And so they all look pretty similar. If all we can see is the density, if all we're going to do is look at imaging, this is what we see. But then if we look underneath and at this phenotypic variation, you can see that they're not all the same. There's quite a bit of variation amongst these. And then if you look at this potential variation of what they could achieve if they had saturated PDGF, there's quite a bit of difference between these cells too. And so what you can see here is, the comparison of these, what we defined as kind of a nodular tumor that's kind of tightly compact, and this more diffuse tumor on the bottom that is got a lot of cells that have moved farther out, and then something in between. You can see that those growth dynamics fit really well. The ratio of cell types are all over the place, as well as, the measured migration speed and percent divisions, and the mean potential, which we can't measure, but I'm displaying there for completeness. And then, ultimately what this comes down to is that because of these differences, you also have differences in response. So now we have applied this anti-proliferative drug to these different types of tumors, and you can see how the average, if you look in A, how the average pans out in gray, and then how several of these other tumors fare. And what's interesting is that there are a few things. If you look at B, C, and D on top, these are things that we can measure at time point 1. So the minute you're going to apply the drug, what might you measure that could be informative? And there's, it's pretty messy here, but one interesting thing is that there is, for these blue tumors, the ones that's recurring and recurring at a much larger tumor, it's got a lot of heterogeneity, and it's actually fast proliferating compared to this red tumor that sometimes recurs and is about to recur at 28 days past the treatment, that has a much less heterogeneity and a lower proliferation rate. We can also look at how it changes between the time point of treatment start and afterwards. And there are some correlations here. I think the most prominent is in the middle there on the bottom, that shows, for some reason my mouse is gone again, that shows basically that you have a larger reduction in heterogeneity and a larger reduction in proliferation rate. So, and you can see in the bottom how several of these tumors pan out, that you've got huge changes in what you start with, and then after treatment, what you end up with this recurrent tumor. Another thing that we observe is that the rim size of these tumors always increases after treatment. So you end up with these more diffuse tumors and less proliferative tumors. And so we can observe how that set of cells kind of lie in this space. If we plot there on the right, the migration in the y-axis and the proliferation in the x, how you start off with a little bit more proliferation, and it kind of moves everything off to lower proliferation rates, because of course we're using an anti-proliferative treatment. One thing to note here, I could never use the word cure. So, Peter Canal, he said, you never use the word cure in GBM, it's called complete response. And so we had to be sure to use the word complete response, because even at this point where you see this red line start to blip up at the very end there, it looks like it had done well, but sometimes it can come back a lot later. And that's why these single cell models really kind of can get to those long-term treatment dynamics a little bit better sometimes than the PDEs. Let's see. So the next thing that we did is not just fit to the size dynamics, but fit to all of the metrics. And you can see there's a lot more, changes in the input parameters over each iteration. So that x-axis is the iteration. I don't think I said that before, but you can see how they change over time. And what we get is a much more narrow group of simulations that actually fit these metrics. And what we're doing here is taking that fit. So we take this kind of best fit that we get from fitting all of the data, and then we say, how does this heterogeneity even matter? Is it worth it to go to this extent and think about all these differences between cells? What if we eliminate that potential heterogeneity and just say that the environment is creating all of the heterogeneity? So you can see on those bottom pictures there that the homogeneous tumor, basically it has all these cells that are exactly the same, and they have the same potential, But then when you measure it, the differences in these cell types are due strictly to the environment, the white and gray matter, and the Pdgf. And what you see is that most of these metrics fit about the same, regardless of whether you have heterogeneity or not. But there is a subtle difference that comes from looking at the distribution of these cell speeds. So if you look at the bottom right there, you can compare the heterogeneous distribution of cell speeds for recruited and infected cells and the homogeneous distribution. And what you see is that the recruited and the infected cells kind of chop off that long tail that you see when that actually is fit when there is heterogeneity amongst the cells. And I think what's happening there is basically that you have Pdgf going from a low amount to a saturated amount, and it basically, you can have cells with a low speed and it hits a high and then just drops off because it can't go any higher. So that long tail is really formed by some of this heterogeneity from having these larger speeds. And of course, it matters ultimately when it comes to treatment. If you don't have this heterogeneity, you're not having selection, pretty much the tumor will respond or it will regrow. So when we do look at this tumor that has heterogeneity, we see that it pretty much grows back most of the time. It only has a complete response 9% of the time, while with the homogeneous tumor, there's a complete response about 76% of the time. So yeah, and one thing that we do notice is that this selection does happen in real tumors. We observed some histology and looked at Ki67 pre and post treatment in humans, and from biopsies, and looked at kind of a similar biopsy in the simulated tumors by measuring kind of an analogous Ki67. And so we found that pre and post treatment, that this Ki67 measure always was higher pre-treatment. Okay. So then we looked at this. We also looked at anti-migratory treatment. So we don't have treatments that actually will kill migratory cells, but there are treatments that will stop migration of cells. And so we wondered, does it help if you apply this anti-migratory treatment? And what we found is that if you look at A here, you can see the anti-migratory treatment is there in pink. It doesn't do so well. And on average, or this one that's displayed here, which is the bolder line. If you look at the anti-proliferative treatment, it does pretty well at the start, and then you get a recurrence. But if you do use these in combination, you do get a response, and it seems to come back a little quicker, but then is a little bit slower for longer term response. And we can plot this down in B as a waterfall plot. So your anti-proliferative treatment amongst this full cohort of best fits, we get this is just fit to the size. We get some tumors that respond pretty well, and a lot of tumors that recur. When you apply this anti-migratory treatment, of course, this isn't killing any cells, so you really just get it to slow down or not. And then when you apply them both together, you also get quite a distribution of different responses. But what happens that's kind of interesting that wouldn't maybe be your first thought is that when you do apply the anti-migratory treatment, you're causing cells to sit still. And so they might still be releasing a ton of this PDGF. So instead of distributing this PDGF out into the tissue, it's just staying still and concentrating more in one location. And what happens is that because of that higher concentration of PDGF, you have a higher response of the cells. So actually by stopping the cells from moving, you're causing this PDGF to build up and actually making their behavior more aggressive. So it doesn't always pan out to apply both of those at once. And of course, what we're seeing through imaging, if all we're looking at is tumor diameter, it's only a little piece of the puzzle. There's still all of these, for the anti-proliferative treatment alone, you can see that you've selected for all these migratory cells. And so even though the tumor appears to be smaller, you have all these cells that have migrated far from where they were. And so it's always this balance of how you're going to choose what's optimal for the patient. And so, yeah, so if we then just look at C there, what I'm showing is that if you're comparing on the x-axis anti-proliferative treatment alone, and on the Y anti-proliferative plus anti-migratory treatment, you can see that really on average, it does better to apply anti-proliferative treatment alone because it doesn't allow those cells to kind of sit in place and build up that concentration of PDGF. And so one thing that we also wanted to test out is there's this idea of go or grow that seems to be pretty prominent, especially in GBM studies. And I think what makes it so contentious is that there's not really a great definition for what people mean by it. One of the definitions means that cells cannot simultaneously migrate and proliferate at the same time, meaning that a cell's not going to migrate and then just split into two as it's migrating. And I think there's no question people don't really believe this. If your cell is moving along, it really only takes about an hour to go through mitosis, and so it will stop during that hour, split into two, and like I said, in this case, they shoot off into different directions following division. So that doesn't matter. I think the more interesting question is number 2, which says, are fast proliferation and fast migration mutually exclusive traits of cells? So can you have a cell that's both good at proliferating and migrating? And I think what we've seen here is that yes, you can. I mean, there's a reason why this is an idea, and it's because you think that there's only a certain amount of energy and you have to divert it in one way or another for a cell. But you always have to look at your environment. And then in this case, we're talking about this growth factor driven tumor. And so there's really no, there's plenty of growth factor to go around most of the time. And so they don't really need to choose one or the other. I think where these trade-offs really happen is when you have a lack of nutrients, a lack of oxygen, and that's when you have to divert from one thing to another. But anyway, we decided to just test it just in case. And so here you can see in the input parameters, there's 2 blank cells. That's because we chose to fit the proliferation rate for the growers, and their migration would be 0, and fit the speed for the goers, and their proliferation rate would be at the low end of the range. And so in doing so, we found that the migration speeds actually fit really well, but then it didn't fit so well on some of the other metrics, such as the proliferation was quite a bit off, and also this distribution between the different cell types. So there was a lot more recruited cells than there should have been to fit well to this. So, ultimately, I wanted to bring this up. It's we've talked a little bit about making a model of a model. Now this is like kind of in the corner of what is relevant for human disease, right? This is really kind of getting in depth into a model of a model, but it does, allow us to take a look at this single cell behavior in context that we wouldn't actually normally be able to observe, and that we'll never be able to observe in a human brain, I mean, because this is really taking this slice and allowing it to stay alive for a little while so we can observe this behavior. One of the things that we're working on is to kind of translate this across species. So they've done this experiment in mice as well, and see that you actually need even more mutations in the mouse to make this occur. You have a lot less recruitment, and it grows a lot slower. And so we think that the human disease lies somewhere in between here. So at least kind of, I think we've been tackling this with an ODE model because it just makes sense to kind of deal with the recruitment aspect of it, and just trying to get at what degree of this recruitment really matters. And obviously, in the rat, this is driven by Pdgf. We didn't consider angiogenesis or necrosis, oxygen, hypoxia, which are all going to influence the selection of these cell types, which really are going to influence treatment. And so translating across these scales and species is really important, and not something that we've forgotten about. And we're also looking at some sex differences that are pretty important in glioblastoma when it comes to treatment response. So just to summarize, some of the main points is that these growth dynamics from imaging alone, you can't use these to determine the underlying phenotypes. They're useful, but tumor dynamics are also influenced by heterogeneity and proliferation, migration, and the microenvironment. And so being able to fit single cell metrics could also help define these phenotypes and help predict response. So we're looking for ways to scale that up into something usable for human disease. And so you can find any of this stuff in our paper in PLOS CB. There's movies on a website that you can find on my GitHub, or the code is on the MathOnco GitHub. Finding either one, you'll find the others. And so, I just wanted to mention, there's so many beautiful, agent-based models. You often get these really beautiful images, and so this work inspired this middle picture here in this book, The Art of Theoretical Biology, where we took part data and part model and fused them together and called that coupled invasion. I also submitted this one on the right that didn't get accepted, but I still really like it. We called Brainstorm. So I just wanted to shout out about that. And this is our group, the Bonk Collaborative. This picture was probably taken in 2013, I think, so it's been a while. We had, a lot of help from, Kristen's lab, Andrea and Susan and Joe, and also Sandra and Luis. Sandy and I at Moffitt Cancer Center. Kristen's now at Mayo Clinic. Russ was with Kristen then, and now has his own lab at City of Hope. And Peter Sanal and Orlando were at Columbia University. So like I said, it's been a while, but this is the core group here. So I just wanted to acknowledge all of them and their hard work. So thanks for listening.
Thanks so much, Jill. This was just a really beautiful talk and like way of building it up. I think it's great, and I really like that model. So I'm really glad to have heard more about it. And on a personal note, I'd be very interested in seeing this Python Jupyter notebook for your hybrid parameter estimation approach. So if you need any sort of encouragement, there you go. So there are some questions, or a bunch of questions in the chat, and I'll try and hopefully we can get through most of them. And if not, of course, we'll do it offline, or I guess online, but off chat. So the first one is it possible to add immune cells in your ABM model to see their effects on the migration and proliferation on glioma cells? I mean, using ABM for predicting the behavior of tumor cells, considering immune cells.
Yeah, that's really interesting. I haven't considered how immune cells affect migration of tumor cells. Yeah, I mean, you can add anything. You know, we made this model the way it is due to the data we had and the questions we wanted to answer. But yeah, of course. And Kristen Swanson, she's continuing to do a lot of great work in immunotherapy and considering the immune system. So yeah, there's definitely room for that. And yeah, they're constantly, yeah, I know Adrian is working on viral therapies and stuff like that that would be useful in the brain context. So yeah, of course. And even, you know, Joe Giuliano, he's got a paper out that we worked on that had to do with the microglia. So the microglia are kind of the immune components in the brain and looking at their detailed movement. But yeah, of course, it would be useful. So.
Yeah, exactly. I think just to follow, Adrian and Paul and I have been working together to on the immune side of it. So I think, you know, it would be interesting at some point to talk all together, because there are obviously different focuses and data from each of these things. So anyway, it's just a follow up there. Miranda Lynch says, fantastic work. The visuals, especially the density plot shown for the cell movement, are so informative. For the movement videos, how are the distances along movement tracks determined? Is that an image analysis approach to determine track lengths for computing the speeds?
So the cell tracking was done by Sanal Nodaswala and by hand. So, I mean, this was a while back, and that's the way they did it at the time. So it was literally taking a mouse and finding the center of the cell. And you saw how these aren't spheres, they aren't, you know, round. So I imagine that even if you wanted to automate it, it'd be a little difficult in this context. But I know that people have done better since then. Even, you know, even 10 years ago, people were having better automated cell tracking. But it does get a little funky when it's really dense, or when, you know, there's a lot of movement. You do lose some accuracy with those automated procedures. But I'm sure if you look, there's something out there. I haven't really looked in a while. But yeah, by hand.
Yeah, it's always I've seen people do that tracing by hand. It's a lot of work, and it's impressive when it gets done. Kathleen Wilkie asks, nice talk, Jill. When you took the top 10% of parameter sets, did I think she's referring to the hybrid approach, did the parameters cluster into local regions, or were the values all over the map? And now my doorbell just went. If all over the map, how did you resample for the next iteration?
Yeah, so, well, what happened was, and I'll pull up this one here to kind of show the input parameters over time. Well, what you can see is that some parameters were quickly determined. So this right here is this consumption rate of the Pdgf. So, you know, what was needed here was a really high amount of Pdgf, and then it needed to be consumed really quickly to be able to make these dynamics. So some of these were quickly determined And then some of them were not really ever narrowed down. So the thing was that I had to do in this parameter estimation is that you would get buildup of error. I mean, more in the, so I'm fitting to these metrics. So you would get buildup of error in a single metric. I didn't really force anything on the parameter side, but just the metric side. And if it got a little too high, I eventually applied a threshold so that there wasn't, just a least total error, but there was also a cap on what a single metric could give as far as error. So if it built up in one metric, it was thrown out. So yeah, and I think this is useful beyond just this one example, but I think there's a lot of refinements, and so that's kind of why I wanted to put this in a nice tool that you can sit there and tweak things, because I think every problem, depending on what you're looking at, it could definitely result in different errors in different places. Did I answer that okay?
I think so, and Kathleen can jump in if she has a follow-up question, perhaps. And I agree. I think having these kinds of tools that then can be built for purpose for whatever you're doing, that's so important, because there's like these different subtleties to each problem. Miranda also asked, oh, Kathleen gave you the thumbs up, so that seems good. Miranda also asked, you can imagine that different configurations of cytoskeletal elements would also contribute to whether a cell would favor division versus mobility. Could this be investigated, for instance, with labeling of cytoskeletal elements?
Yeah, that's interesting. I think they might do that already. And I think some of the anti-migratory drugs target cytoskeletal, rearrangement, or I don't know exactly, but yeah, migration is an interesting thing to study, and it's not necessarily like easily observed unless you are digging down into this single cell stuff. Well, I mean, they do it in in vitro, but not necessarily that much in vivo. But yeah, I bet that would be a nice stain. I don't know what else, but yeah, there's all sorts of stuff involved in migration. Yeah, that they could put a tag on and watch it and where it ends up, because it's really, I mean, when you think about, like, you look at a single snapshot of a tumor and you have these invasive edges, it's not always what you think you're seeing is what actually happened. You do have proliferation that causes invasion. You have proliferation that's causing the growth, but you also have just straight invasion. And then you have like leader and follower cells. So you can have all these projections, and it's not necessarily strictly from invasiveness, but it's also this combination of proliferation and migration. Mm-hmm. So that's why I like these two traits so much, because there's so much emergent behavior underneath it all that's not entirely intuitive until you start digging into it.
And speaking of that, I know you said in the 2 points of migration and proliferation that the number 1 was maybe less interesting, but I was quite interested in it. And I was interested in what your thoughts are about why, after cell division, a cell would shoot away. Do you have any, I was trying to think of a rationale for that, but I'm not sure.
I don't know. It's just what they observe. Maybe I don't know. It's just I have no clue.
Yeah. You don't have to have an answer. That's okay.
Yeah. So yeah, no, it is interesting, and but I think it's an obvious, like, you could see why it wouldn't be moving for an hour or something to just go through all the cytoskeletal rearrangements to be able to split into two.
Yeah.
But we're talking about an hour out of like maybe an average of 50 hours. So it's not really like in the grand scheme of like the larger time scale of the dynamics. It's just sitting there for an hour.
Yeah, it's kind of insignificant, right? Well, thank you so much again for your talk. Just really, really beautiful work. And we're so glad that you were able to share with everyone.